This is episode 108 of the Immunology Podcast, the Intestinal Epithelium, with Dr. Mahima Swamy. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Ratt. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. And if you enjoy the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Mahima Swamy from the University of Dundee on the podcast to talk about her research on immune interactions at the intestinal epithelium. We also got our usual roundup of recent highlights in immunology news coming up. But first,
another reminder that IUIS 2025 is coming up. We can't wait to meet you in Vienna. Be sure to check out the program and register at iuis2025.org, and we'll see you there.
All right, Brenda. T cells in the gut.
The best of both worlds, isn't it?
It was amazing.
I have to say, I mean, obviously I win because that shows that T cells are everywhere and they're everything.
But you already know that T cells are everywhere. That's not new.
Well, I mean, it's always nice to talk about the gut, and I guess that
it's kind of the front line of the immune system, that and the skin, right?
Indeed, probably more than the skin. I think it's true. The gut is
the gut's a little dirtier than the skin, unless you're showering very well.
There's nothing I can say. So I think you should hear it from Dr. Swamy herself in our second part of the show today.
Oh, absolutely.
But before that, what do you have for me? What's the science?
Fungus among us all day.
Oh, fungus. Okay.
Today is fungal day. Fungi, fungemia, fungal fun. All right. So this is in Cell coming out July 24th.
That's in the future.
Well, the paper's here now. Phenotypic landscape of an invasive fungal pathogen reveals its unique biology. All right. First author is Michael J. Boucher. Last author is Hitin D. Madhani. And there are no extra first authors on this one. I'm 99% sure. I ran out of superscripts. There we go. So, Cryptococcus neoformans is actually a pretty major cause, the most common cause of fungal meningitis, like when your brain gets in your meninges, not actually your brain, but the tissue lining your brain, which is usually where you get infections, gets infected, and is the top ranking WH fungal priority pathogen. So this paper is going to be actually pretty quick to describe, but also super cool. They studied C. neoformans because it has conserved eukaryotic mechanisms lost from more specialist model yeast lineages, right? So what we usually study with yeast in Petri dishes, a lot of those have lost things that are common eukaryotic pathways for a variety of reasons. So they do a knockout gene deletion library, 4,328 gene deletions, and measure the fitness of each mutant under 141 growth-limiting conditions and during murine infection. So, like, just the amount of work here and the multiplexing is crazy. And so they had learned kind of what a lot of these genes do that weren't previously described, and they identify environmental adaptive mechanisms that are important for virulence in mammals. And they kind of show how you can do this and then provide a resource for this. So at a high level, I'm kind of done with the paper because like it goes on about 15 different mutations-ish kind of sorta and talks about a whole bunch of stuff that if you're not a Cryptococcus person is not super interesting. But the fact that they can take an entire genome and do what they did here, that I think is really interesting. Right. So they used a biosilic transformation, which is where they insert the gene on like a microchip that like I had to look this up. They use like this metal surface to use it to inject the genetic code that causes a knockout. And then that's a way they could really multiplex this. And they did a bunch of sequencing to verify it. And then they did a whole bunch of fitness screens. And so part of this biosilic thing, I'm like, well, that's kind of neat. I haven't seen that before. And that was cool. And then they find some new insights into a hedgehog-like pH sensing pathway. And part of the ability of these pathogens to survive is being able to tolerate 37C and high pH, because fungi tend to be a little finicky, like things a little cooler. So there is that, which they get into. So they identify this hedgehog pathway called RIM 101. They go and they look at calcineurin signaling and find some effects there. And and that's, interesting from the perspective of pathogenesis there, and controlling resting membrane potential to tolerate pH. They find some non-homologous end joining repair, important pathways and new genes there. And they look at fitness genes. And again, they find a whole bunch of stuff with fitness that really affect its ability to tolerate pH and temperature. And then hyaluronic synthase was important for virulence. And then they found some inducible factors as well. So part of this, gets in the weeds that I don't even appreciate, but fundamentally they're able to map out new genes that were important at different varying temperature stresses and at different pHs that weren't known before, which means new drug target. But they're also able to solve some of these have homologs to human or other mammalian genes that don't have a known effect. And so they're starting to map that out too from other signaling. And it's really more to me about a tour de force of being able to nail down everything and its uncle about one organism. And immediately when you do that, you start finding new cool things. I'm like, oh, okay.
How many genes does an organism like this have? Because we have 30,000, is it us?
Well, I do not know the total number, but they did 4,000 different deletions. The total predicted protein coding genes is 6,975.
So wait, then we cannot have 20,000, 30,000. That makes sense.
No, we're at 15,000 or something.
15,000.
I thought. And so obviously some of these and so they said they're purported about 1465 essential genes. So if you knock it out, it dies. So they had about 79% coverage of non-essential genes. Obviously, if it's essential and you knock it out, you get dead organism.
Not much to study there. Well, it's cool to see. So I guess their knockout, is it with endonuclease, Cas proteins that they're doing it? Or how is it that they're knocking out these genes?
That's with this biosilic transformation, where they use the silicon-based thing to insert the genetic homologous flanking region, and then it homologously recombines and knock it out.
Oh, okay.
They just use the silicon-based knockout approach.
Oh, that's very interesting. Oh, I don't know a lot about fungal gene engineering. Yeah. All right. Okay.
But it's a way of so it's a homologous recombination-based knockout. And I think there's a neomycin cassette or something in there that it swaps it for, so you can select for it, I think. But that they do a knockout with the flanking regions.
All right. Very interesting. Thank you for sharing, Jason.
Fungus.
Jason Fungus. Okay. So I don't have a good segue here. I want to talk about gamma delta T cells, because what else am I going to talk about? So this paper,
gamma delta kind of year.
Yeah, I mean, they're having a comeback. They're having a renaissance. All of a sudden, they're everywhere. So this paper, I think, is really cool. It's a very cool structural paper. And also going about, rigorous observations to understand. So the paper is called Structures of Butyrophilin Multimers Reveal a Player-like Mechanism for V-GABA 9 V delta 2 T cell receptor activation. And clearly it was a work in big collaboration. We have 4 first authors, Mai Zhang, Yi-Ping Wang, Ning Kai, and Ying Ku, from corresponding authors Yunju Shao and Yongshui Zhang. Oh
You did
man,
a good job murdering just like I do. Welcome to the club.
I need to get, if there's any of our listeners, any of our Chinese speaking listeners who like to give us a little instructive on how to properly pronounce these names and honor them as they deserve, just let us know. Just reach out to us. All right. They are Peking University and Tsinghua University in Beijing. And basically, this paper, as I mentioned, is a very kind of structural cryo-EM tour de force, in which they're trying to understand how does the gamma delta TCR work. Because we kind of understand how the alpha beta TCR, recognizes peptides in the context of MHC molecules, but that's not what gamma delta T cells do. So the most well-known ligand for gamma delta T cells is this molecules, these proteins called butyrophilins, BTNs, so which are transmembrane proteins that can recognize phosphoantigens. So they recognize phosphorylated proteins inside the cell, and they kind of somehow they are transmitting this information out for the gamma delta T cells to see. And it's important because these phospho antigens, quote unquote, they are usually non-peptidic metabolites that are often associated with bacteria or parasites or some dysregulations also in cancer. So basically they're one way of looking into the cell. This clearly this metabolites are very important and very informative, and we have developed a way of seeing them. So when it comes to these BTN molecules, usually I think at least in the case of BTN molecules that are interacting with V gamma 9 V delta 2 TCRs, they are composed of 4 BTN molecules, a homodimer of BTN 2A1, and a heterodimer of BTN 3A1 and 3A2. And all of these molecules are necessary to get recognition by gamma delta T cells. So the question is, how are these binding? There's no MHC. Clearly, this is not how it's going. And there's still, because it's difficult to really characterize these structures because, they're flexible, because they depend on whether they are binding to their ligand inside the cell, whether they are binding to the gamma delta TCR. So what they clearly, after probably took a lot of work to get these structures really worked out, the authors were able to compare cryo-EM structures of the BTNs bound to their intracellular domain, to their ligand inside, and also when they bind to a gamma delta TCR, trying to understand what happens to this structure. And what basically, long story short, what they see is that there are these structural changes that happen that allow the complete binding, the kind of the functional binding of the BTN extracellular domains to the TCR. And basically, the structure that you see, they have some really nice kind of models in which they bind to the TCR is, they say, by like a plier. So basically like a little, like a little plier holding the TCR from kind of both sides. So one side being the homodimer, the BTN 2A1 homodimer on one side, and the heterodimer on the other side. And there is when this binding occurs in kind of stages, and you have in order to have a complete binding, you have a complete kind of change. There's these domains that are at the end of the extracellular domain of these molecules. So usually the BTNs are kind of binding to each other. The two dimers are interacting with each other, but when they come in contact with the gamma delta TCR, then this space becomes available. There's a conformational change in the TCR molecule, and this allows the TCR molecule to bind basically at two parts. They bind on one side. If it's they are there's this previously inaccessible area of the BTN molecules on the surface of the BTN32A, so part of the heterodimer that gets this conformational change that allows the binding to the TCR. So it's really a dynamic interaction that happens between these two structures. And basically what you see in the end is a structure in which the B delta 2 binds to the BTN32A, and then the homodimer of the BTN2A1 can access the delta, the end of the delta, a part of the delta chain, and that's how you end up having these little arms that just grab the TCR from these two parts.
I know the audience can't see this, but you could do it in hand puppet for me.
Yeah, I'm sorry, dear. I'm trying to it's Jason is seeing. I'm just kind of trying to show you my hands, but basically, what would it be if you're like just grabbing? Imagine you're with your two hands. You're like your fingers, together, and you're just like cupping the TCR with both your hands, kind of slightly different angles, but that's how kind of it grabs, grabbing the BTN molecules are grabbing the gamma and delta chains from at least this particular TCR. There's different V and gamma delta chains, so I'm not sure if this applies to every combination, but V, gamma 9, delta 2 are pretty common. One of that was the most commonly studied gamma delta combination. So it's really interesting. Of course, there's a lot of really cool images, a lot of structural analyses, measurements, understanding how different molecules or antagonists or agonists work, and what are the residues that are forming part of this interaction. So that's very in the weeds, but in general this is really nice because I don't think we really we had a really good understanding of how is it that this interaction takes place. And you never know, this might help us guide some kind of other molecules that are affecting gamma delta functionality, if that were something we wanted to do.
So as someone who usually does the structural biology, I appreciate you doing this. I think it's always important to understand how it goes.
Yeah. And it's in the end, the gamma delta TCI is such a fundamentally different animal from the alpha beta. The fact that they're both called T cell receptors feels a little bit weird because they are recognizing completely different things in completely different ways.
All right. Well, I guess it's my turn now, right?
Yes.
Guess what I'm going to talk about?
Fungi.
Yep.
This was fungal week.
Fungal week. It's like Shark Week, but grosser.
Mushroom week.
Yeah. All right. This one is in immunity. Detectin-1 facilitates lung fungal mediated pulmonary fibrosis. First authors are Ding Qiu, Shixin Zhang, Shanyan Huang, and I'm done. Last author, corresponding authors, there's 3 of them, Shui Wang, Xiaowei Dong, and Sei Dong, from a whole bunch of institutions that I will not attempt to butcher. Comes out July 8th, so also in the future. So what is this one about? So Detectin-1, whose gene is CLEC7A, is a C-type lectin receptor for beta-glucans. It's known to be important in host fungal infections and has been implicated in allergy responses. This paper looks at pulmonary fibrosis. And one of the ways you do pulmonary fibrosis in mice is you give them bleomycin, and that induces this macrophage-mediated pulmonary fibrosis. And pulmonary fibrosis is a big bad thing in humans. We can't treat it very well. It's sometimes idiopathic, sometimes caused by drugs, sometimes caused by disease, infection, leads to bad news bears, lungs get all fibrotic, then they don't work very well. So not good times, right? So they're trying to understand what this really does. So they know it's predominantly expressed in alveolar macrophages and that it correlates with fibrotic severity in human and mice, and they kind of reestablish that trend here. But they interestingly find out that when they knock it out, you have less fibrosis. And they try to figure out why. And so what they did is they sorted out that at a high level, if you knock it out, you have less arginase 1 and TGF beta producing alveolar macrophages and less overall profibrotic factor expression that's already known. So then they're like, well, is this because other macrophages are coming in? What's happening with the alveolar macrophages? So they do a bunch of tissue specific or not tissue specific. They do a bunch of transgenic knockouts where they do where they use the CD45-1 and CD45-2 pairings and then cross promote, cross transfuse one of those into the other host, right? So they'll take a CD45-2 Declin-1 knockout or CLEC7A knockout and put that into a into a mouse whose alveolar macrophages have been depleted or not, or whose peripheral macrophages have been depleted or not. And then they'll do lineage tracing as well and look at that. And so what do they find? So they find it's not some other random macrophage stuff. What it is that it promotes, Dectin-1 promotes quiescent alveolar macrophage differentiation into pro-fibrotic macrophages, and they identify that as being part of it's RAF1 dependent, and it bypasses card signaling and monomacrophage chemotactic recruitment. It's all very local to the macrophage itself, the alveolar macrophages. And so that RAF1 then goes and is a transcription factor, leads to arginase 1 and MMP and TGF beta. But it's all local there. It's not some cross-promoting, causing something else to come in or out and do it. The alveolar macrophages differentiate differently. And then, you didn't think I was going to do this without doing the microbiome, right? No, yep, all right. Got the smile. So, fungi are part of the microbiome too, or the mycobiome as it's sometimes called. So, they have found that if you ablate commensals, especially its commensal angiodontium, the effect goes away. So you also get you get this, you ablate the bleomycin causing it. And so what's happening is that Dectin-1 is detecting commensal lung fungi in the setting of an injury, bleomycin, right? Which causes lung injury. And that then leads to the alveolar macrophage undergoing differentiation to a fibrotic state and causing fibrosis. And that's basically the story. And they map this all out. So the alveolar macrophages are the culprit when they detect commensal microbiota in the face of injury or fungi.
So is this macrophage is going rogue. What's
The
up
T
with
cells
them?
never go rogue.
Never.
No
It will never
disputing.
betray us.
None.
Famous last words. Yeah, but they can very easily confuse macrophages. Yeah, but I don't know.
They're just hungry and they just start eating. Can you really blame them?
I guess not. They're just responding to the signals that they get.
Mm-hmm. Aren't we all?
I guess. Well, all right. So for the last paper of today, I have a very interesting story, very relevant. Although it's one of those stories that I also was always important to take, with a grain of salt and then think what are the real implications. But it's kind of a thing that you would have that would be suitable for some big news, like your headlines. That's the word I'm looking for. So the paper is called "Early Life High Fructose Impairs Microglial Phagocytosis and Neurodevelopment." Kind of scary. Published in Nature on the 11th of June. And first author, Zhao Quan Wang, and from the lab of Justin Perry at Memorial Sloan Kettering Cancer Center in New York. And basically, I think the title already says a lot, but what's important? What do we need to know in prepare to understand this paper? So first of all, I'm no neurologist, I'm no neuroscientist, but I do know that there is that there's a very critical part of like early development, neonatal development, and childhood development in which there is this process. I don't know, is it correct to call it pruning?
It's
But
called neural pruning, where you pinch off a bunch of connections and kill some of your neurons so that you have
yeah.
right amount of connections. And people who have too many, that's one of the thoughts behind schizophrenia, is that they're connecting things that should not be connected in a developed brain.
All right, so that makes sense. Also, there's this other word called phagocytosis, in which I think it's more than pruning; it's more like really clearance by phagocytosis of dying neurons. They are dying as part of this kind of reduction in neurons that happens. And for this particular process, microglia are very important. This, no, talking about macrophages, these resident macrophages that live in the CNS, and they are kind of really important in eliminating dying neurons that die around early development and are important for correct brain structure. So there's that. And there's also the fact that for some reason, which we're going to talk about later, there is adults express quite some fructose, high affinity fructose transporter called GLUT5, also known as SLC2A5 for those working in immunometabolism or metabolism in general. And interestingly, so these are kind of one of the few cells that expresses this high affinity transporter. The only immune cell that expresses GLUT5. So it is expressed in kind of the gut, but the only immune cell that's really has been shown to express GLUT5, this high affinity fructose transporter, are microglia. And but a little bit less so in neonates in early life, but then they get it. It looks like expression is increased in adults. So that's kind of what we started with. And of course, one of the things, sugar and what's sugar doing to us. And I think this is always a question. And they are trying to look into the effect of fructose, of high amounts of fructose in neuronal development. So basically, what they take a few unlucky neonatal mice, they inject intragastrically fructose, so they give us a high dose of fructose, and basically what they see is reduction in the number of microglia and increased numbers of uncleared dying cells in this neonates treated with high fructose. And I think they look at particularly in the prefrontal cortex, because it's one of the early stage is one of the main proportion, the main proportion of brain volume is in the prefrontal cortex. And it's also important because we're thinking about behavioral effects, makes sense to look in there. So they also see that if they have, if the mothers that are either, gestating or have given birth or giving milk to neonates, also affects the brains of the offspring at their young age. And basically what they see is fewer microglia and more dead cells. So like the microglia are not doing their work. They're not being phagocytic and getting rid of those dying neurons that are part of the normal development of the prefrontal cortex. So they do a lot of different experiments around, giving them at different points, and they also start looking at specifically the importance of GLUT5 in this effect. And they see that indeed GLUT5 is expressed on this microglia. They show it in their own data, and that it is important for transporting the fructose inside the microglia. So microglia exposed to fructose that are expressing GLUT5, then that really allows the fructose to come inside. And basically, they also have experiments with mice that don't express GLUT5, knockout mice, and again, they show that this effect is very much due to the expression of GLUT5 on the microglia. And if you remove the GLUT5 in the microglia, you don't see this anymore. You don't see this effect. You don't see this reduced phagocytosis and this kind of loss of function of the microglia in the young mice. Interestingly, is that they do some behavioral studies and they show that to some extent there are certain things that the mice they're already seeing, like as these mice reach mouse adolescence, they perform a little bit worse in what is called the fear extinction protocol, in which they basically they see how long they take into forgetting something traumatic. Basically, I forgot what it's exactly that they do to this poor mice. They are a learning task in which they're conditioning the mice, and then they wait to see if the mice forget this, or they get kind of they don't become anxious from this behavior. And basically the idea is that if you cannot get rid of that anxiety, that is a misadaptation that you have, so that you're supposed to be able to come and calm down. And this is basically what they see, I think, when it comes to the main effect that they see behaviorally. That's it. I had to say that sometimes these behavioral studies in mice is a bit difficult because of the things that they do. But basically, they show on the one hand that they see more dead neurons, that they see differences in the phagocytosis of the microglia. There are probably behavioral effects on this, and that this is depending on GLUT5 expression. And I would say that it's still, I think the effects is pretty clear. I think the fact that the microglia look different and they're not behaving normally, I think this is really clear. They show that in part this is because of the inhibition of, no, the enforcing of these hexofructokinase that is part of the downstream of the metabolism of the fructose, and this is part of the mechanism by which this seems to be impairing the function of this microglia. But I would like to see, I think it would be really nice to see, hard to see an experiment in humans, but I wonder how much of this can be really then later applied to human behavior. But definitely we are consuming more fructose than we used to. And I wonder whether that is affecting kind of children's behavior in the long term. This is what they seem to be suggesting.
So you could do a natural experiment, because there's people who have GLUT5 deficiency and are fructose intolerant. So when they eat things like onions or other high fructose foods, they get a big old tummy ache. I happen to be one of them. So you could compare them, who can't have fructose, and you don't have fructose transport unless it's in equal amounts of glucose, because or sucrose, because then there's a dual uptake for fructose plus sucrose, but just the regular fructose uptake, no bueno. So you could do a natural experiment that way.
Yeah. It's funny because they don't even mention that in the discussion.
It's super not really known. Or like, it's a known thing if you look, but if you don't look, you won't know.
Huh. Yeah, it would be very interesting to see. Definitely. It is very intriguing. We're becoming such a direct connection. I mean, I was not expecting something so direct.
Well, what's also going to be interesting is our talk with Dr. Mahima Swamy at the University of Dundee in just a moment. But before we get to that, whether you're working on autoimmunity, infectious disease, or immune regulation, Stem Cell Science News delivers the latest research directly to your inbox. Subscribe to one of their 20 immunology and cell biology newsletters by July 18th, and you could win a personalized lab coat. Visit stemcellsciencenews.com/giveaway.
Well, for the second part of our show, for an interview, we are very happy to welcome Dr. Mahima Swamy. She is program leader at the University of Dundee, and her lab, basically, most of his lab research interest is around understanding very special immune cell type, the intestinal intraepithelial lymphocytes, which are this kind of unconventional set of T cells that I'm very curious about. And I think this is a double, kind of a double whammy for us because it's in the intestine and it's T cells. So you're going to have both of us, Jason and I's attention for today's discussion. So Dr. Swamy, thank you so much for joining the Immunology Podcast.
Thank you. I'm really excited to be here, having been a listener of your show for many years.
Thank you for being here. All right. I'm going to jump in because Brenda had a T cell rampage with the last few guests. What makes an IEL, which is short for intraepithelial lymphocyte, but we're going to say IELs because otherwise we're never going to get through talking to each other, different from a regular T cell?
I think the biggest thing that makes them different is that once they go into the epithelium, they stay there their entire lifetime. So they don't really have a chance to meet an antigen-presenting cell anywhere.
All right. But then what's the T cell without antigen presentation? How does that work?
That's the $1 million question. We don't really know. There's still really no proof that IEL use the T cell antigen receptor to respond when they're responding to infections or stress in the gut.
So maybe for those that don't have the structure of the guts in our minds and all the different layers, how can it be that there's a place that their antigen presenting cells don't access? How, when you think about all the layers in the gut, where exactly are we finding these T cells, and what other kinds of T cells might be around, but they're not IELs?
So when we think of the gut, it's actually quite amazing that there's just a single layer of epithelial cells covering the entire surface of the gut. And within that epithelial layer, 95% of the immune cells that you find are the T cells, and these are the intraepithelial lymphocytes. Below the epithelial layer, that is below a basement membrane, there are lots of other immune cells in an area called the lamina propria. And there you will find macrophages, dendritic cells, B cells, and a lot of innate lymphoid cells as well. The dendritic cells and macrophages do extend processes through the epithelial layer, and they can talk to the lumen of the gut. But there is no evidence to date that they actually talk to the IEL. And there are a lot of IEL and much fewer of these other cells in the epithelium.
So with these IELs, once they get in and they stay there, what do we know that they're doing if they don't, play around with antigen all the time? What are the main functions that we see they have?
So what's really amazing is that a couple of years ago, there's been 2 studies from Karen Edelblum's lab as well as Daniel Maceda's lab, and they show that IEL are really zooming around the gut. So they're constantly patrolling the epithelial layer and checking, pretty much getting in contact with every epithelial cell once every 8 minutes or so. And so that means that they basically seem to be talking to the epithelial cells and not really to any other cells in the gut, as far as we can tell. And what's really cool is that they also found that in the context of infection of the epithelium, these epithelial cells stop moving. So if they stop moving, the idea is that they're responding to infection of the epithelium. But we don't know exactly how they're stopping yet. And we don't know what the signals are for them to stop. And that's, I think, an area we're really interested in looking at. But what we could show from my lab was that they can kill the infected epithelial cells really rapidly, and they help to shed them into the lumen. So you can get rid of the epithelial cells that are infected really quickly.
So you're telling me that epithelial cells in the gut are interacting with their immune cells, like if they were themselves immune cells? Oh no. This has been
how does that feel, Brenda? I've been telling you that immune cells are just basically or epithelial cells are super macrophages.
No, I refuse to believe it, but now the evidence is, uncontestable and undeniable. Okay, so what are the mechanisms that we know that this communication goes? What are kind of the established, yeah, cytokines or markers or molecules that we know are intervening in this conversation?
So we know that epithelial cells already have a whole set of molecules that allow them to respond to infection or to stress. And so when they do that, then they give out something that must be sensed by the immune system, be it macrophages or IEL. And in the context of IEL, one of the most important cytokines we think of is IL-15. And IL-15 is a really interesting cytokine because it's transpresented. So it's actually kept local. That means that the IL-15 receptor alpha chain is on the epithelial cell, and IL-15 is directly presented by the epithelial cell to the IEL. And this seems to be a kind of a danger signal. So when the IEL sense IL-15 on the epithelial cells or high levels of it, then they sense that something's wrong with the epithelium. And we found that this signal is really, really potent signal for IEL activation. So they can respond and they can kill and they can proliferate. And while otherwise they seem really quiescent cells, just basically monitoring, the moment they see IL-15, they go a bit crazy.
So then I imagine if you have a local immune cell that can talk to epithelial cells and then kill them, that they could be involved in the pathogenesis of things like IBD, right?
Yeah. Yeah, I mean, it's a question that I've asked, looked around a lot for the literature for any evidence that exists. And the evidence has always been kind of correlative, that we can see that there's increases of these cells in certain diseases, in Crohn's disease. But it's not really obvious what exactly their role is there. However, in celiac disease, which is an autoimmune disease that also causes damage to the gut a lot, there's a really, really clear correlation. So we know that in celiac disease, in the response to gluten, it's the CD4 T cells in the lamina propria that recognize the gluten, but it's the IEL in the epithelium that go around damaging the epithelium and completely destroying the epithelium, which is the autoimmune component of celiac disease. So what they're doing in IBD, we don't know.
So in celiac disease, how do the CD4 cells tell the IELs to go kill stuff? Or is that not what's happening?
I think they must be, but one cytokine we know that's important is IL-21 that the CD4 T cells make. CD4 T cells in the ones that recognize gluten can also produce interferon gamma, which would damage the epithelium. And that damage is enough to then cause the epithelium to produce a lot of IL-15 and also stress signals, which then seems to give like a feed-forward loop telling the IEL, okay, things are going wrong. I'm not well, get rid of me. And so then there seems to be a lot of damage in the epithelium. And when you look at images of a full-blown celiac disease, when an actual reaction is happening, you see there's more IEL than you can see epithelial cells. You hardly see any epithelial cells, but that layer is full of IEL. And that, I think, is just really amazing, that how we can even survive with completely damaged or destroyed epithelium.
Wow. Because just to state the obvious, T cells don't make IL-15. So that activation has to come from, for example, other epithelial cells are the ones that are making. Are there other sources? What are the main sources of IL-15 in general?
Lots of cells can make it. So dendritic cells can make it. Epithelial cells can make it. Macrophages make it. Lots of cells can make it. I mean, many stromal cells also can make it. So we don't know exactly where, but because of this transpresentation, it's because it seems to be tied to the surface of the epithelium. And so the cell that's most likely to see it is the one that's sitting right next to it that has the IL-15 receptor chains that can recognize this transpresented IL-15.
Right. Exactly. Because it's tethered to the cell. There's not just released into the matrix.
Exactly.
There's a few cytokines that do that, right? They're not like we think of the cytokines as, just being released, but like TGF-beta as well is often found kind of bound to the cell. They're not necessarily just floating around. whatever fluid it is. Sometimes it's hard for me to think about, these things that we work so much in culture and just add it to the media and think,
Yeah, exactly.
that's how it works, right? But no, actually, there is this whole structure, and these cytokines are not just released, nilly-willy into the environment.
So then I know to kind of get to the other side of cytokines and signaling, you also really have started to focus on some of the specific signaling pathways in these IELs, right? That's LRRK2. Could you fill us in a little bit about that, what that kinase does, what it's, what it senses, and then how it's behaving in these cells?
Okay, actually, the cytokine that is most interesting in the IEL is the PIM kinases that we've been working on, because we found that in the context of IL-15, the PIM kinases really seem to be master regulators of IEL killing and proliferation, and their ability to grow. All of it seems to be like they're master metabolic regulators of these cells. LRRK2, which I've recently been publishing on, is a kinase that's actually not expressed in T cells. It's expressed in a lot of other immune cells: B cells, macrophages, neutrophils, dendritic cells. In fact, every cell except a T cell, it looks like. But the reason we got really interested in LRRK2 is that it's mutated in Crohn's disease as well as in Parkinson's disease. And what's really cool about that is that there's this idea that Parkinson's disease originates in the gut. And the fact that Crohn's disease might have a similar basis as Parkinson's is really interesting. And so that's why we started looking at these mutations and what they might do. And what we're finding is that there's a lot of dysbiosis created at the gut when there are mutations in LRRK2. And also, what we find there is that IgA production by B cells seems to be regulated by LRRK2. So we think that there's a connection there. And this is just really, really new work. So we're trying to figure out exactly how this might be connected with Crohn's disease or with Parkinson's disease.
So it's interesting that you mentioned Parkinson's disease and, of course, the work that you've published. But I would just like to take a step back and go back to that, because we've already also covered in the podcast certain papers in which, things that happen in the gut get transported somewhere else. And then you have, responses to microbiota in the gut affects the lung affects. Can you maybe explain for our listeners that are not aware of this? I think this is kind of a surprising observation, and that there is this connection between the gut and development of Parkinson. What are the mechanisms that we think are playing, and what is that people are seeing that makes them make this statement?
So I think one of the things that is really interesting is that they took the fecal material from people who have Parkinson's disease and transferred it to wild-type mice. They found the mice develop Parkinson's-like symptoms. So that was the first really, I think, interesting connection between maybe something going on in the gut and something going on in the brain. But there's a lot of data. So Parkinson's disease is caused by aggregation of alpha-synuclein in dopaminergic neurons, which causes death of the dopaminergic neurons in the brain. But we don't know the exact sequence of events and what exactly is happening. But there's an idea that actually long before people develop Parkinson's-like symptoms, you can already find aggregates of alpha-synuclein in the gut. So maybe it's being seeded in the gut and then it's being transported to the brain. And then there's this other connection, which is that people who have IBD have a much higher risk of Parkinson's disease. On the other side, if they have their IBD is under control, so if they are on anti-TNF therapy, then the Parkinson's risk goes down to normal. So like the general population. So there are lots of smoking guns, but there's no one clear connection that, okay, this is definitely happening in the gut.
Have they done anything with antibiotic ablation or anything, or looked at antibiotic exposure as it relates to Parkinson's?
There's some correlative studies, but it's not really clear whether that's happening. One of the issues with Parkinson's is that you develop symptoms a lot by the time you develop symptoms,
You're
things have
way
gone pretty
past.
bad. Yeah. So whether, maybe it would have made a difference if you'd done it much earlier, but then the question is, how do we know that this person is going to get Parkinson's disease?
I just want to take a step back and go back a little bit to the IELs. And we already kind of touched upon what they're doing, where they are present, and how they're different from, for example, cells in other parts of the gut. But how do they get there? Do we understand at which point in development do IELs populate the epithelium layer, and where are they coming from? A specific population, or they're just T cells that are around and got kind of coerced to become IELs?
So yeah, I think this is really important, that many people think of IEL as gamma delta T cells, but they're not gamma delta T cells only.
Right,
In
they're
fact,
alpha
in
beta.
a human, it's like 90% alpha beta T cells and only about 10% gamma delta T cells. So where are these alpha beta T cells coming from? With gamma delta T cells, we know that their development is slightly different, and they seem to be developmentally targeted to the gut. But what about the alpha beta T cells? So it looks like there's two populations there. One set of alpha beta T cells seem to be responding to gut antigens, maybe microglial antigens, normal food antigens, whatever, that are normally present in the gut. And then they go and see the epithelium as a kind of memory cells that are sitting there. But they have gone through normal thymus development. But the cells that I'm really interested in there's another subset which are called natural IEL. And these are alpha beta T cells that go through something called agonist selection in the thymus. So agonist selection is when you have a strongly self-reactive T cell antigen receptor on your surface. And normally those cells should get deleted. But the ones that seem to have the highest autoreactivity are somehow transformed into IEL. And so directly from the thymus, those highly self-reactive T cells become IEL, TCR alpha beta, CD8 alpha alpha IEL in the mouse. And they're also present in humans. And the question is, why do we have self-reactive T cells in the gut? And we don't really know why, but there's this really important population that sits there that is directly targeted to the gut.
Have they knocked them out to see what happens in mice, at least?
There's no way to knock them out specifically because they're alpha beta T cells.
Roll right in. They then had something that's an IEL specific that you could poison or something afterwards and knock them out.
Yeah. So it's something we're working on. But we've not managed to knock out only these unconventional alpha beta T cells. What we have managed to do is now make a mouse model where we can knock out all IEL. So that includes the gamma delta T cells and the alpha beta T cells, the normal conventional alpha memory-like tissue resident memory type T cells, as well as these natural self-reactive alpha beta T cells. And what we're seeing with that is that the epithelium is just changed completely in the absence of these T cells. And that's really, really fascinating to us. We're seeing that basically all their nutrient uptake pathways are completely changed in the absence of these T cells.
So just to make sure we're extra clear, there's so many interesting non-conventional T cells. You got your IELs, which have a TCR but don't move. Then you have your innate-like, which don't have a TCR, right?
Mm-hmm.
And then you have resident tissue memory T cells, which are T cells in the tissue, but are not IELs. So how are resident memory T cells not IELs, but also in the tissue?
So they are IEL too. According to me, they are IEL too. As long as they're intraepithelial and they don't leave the epithelium once they seed the epithelium, they are intraepithelial T cells or IEL. So that means that includes tissue resident memory cells. That includes innate lymphoid cells maybe as well, but those are a very small population. And then we have the gamma delta T cells, and we have self-reactive alpha beta T cells that go directly into the epithelium, which are called natural IEL. So there are lots of different populations there.
How is it that you test that these cells are self-reactive? How do you really evaluate that self-reactivity?
So this was done more than 20, 30 years ago now, where they showed that, when, like, for example, the HYTCR, which is a transgenic TCR, where the antigen is normally expressed in male mice and not in female mice. So normally in male mice, there's negative selection and all the T cells are gone. But if you look in the gut of male mice, of HYTCR transgenic mice, they still found alpha beta T cells in the gut. And when they looked at these more closely, they express CD8 alpha alpha on their surface. They don't express CD4, they don't express CD8 alpha beta, but they express CD8 alpha alpha, and they're in the epithelium. And so there was the first clue that these cells are self-reactive. So they should have been deleted. They're gone from nowhere else in the mouse you find them, but you find them in the epithelium. And then since then, there's been a number of studies where they cloned the TCRs from mice from these natural populations, and they found that, again, that they are self-reactive. They've also shown that if you make transgenic mice with these TCRs from the gut, those cells will only become IEL. They won't become any other cell.
But is it common then to have this unconventional co-receptor structure, alpha alpha, or is that like a requirement to have the self-reactive to not have the proper co-receptor distribution?
I think it is, because actually the idea is that we don't want them to react to their TCR anymore because they're self-reactive. So CD4 and CD8 alpha beta help the T cell receptor to recognize their ligand. But in this case, these T cells, actually, we don't want them reacting too strongly with their self-ligand that's present in the body. So that's why they seem to express CD8 alpha alpha and not CD4 and CD8 alpha beta. The exact function of CD8 alpha alpha is not clear because, again, you can't knock out CD8 alpha alpha alone. It's kind of impossible. But the CD8 alpha alpha does seem to, when you overexpress it in cells, it seems to have a negative signaling role. So it actually turns off signaling more and is immunoinhibitory to TCR signaling.
Well, that's a very good point, because maybe I would say self-reactive, like self-aware, but because you don't have, for example, this enhanced LCK recruitment that usually happens because of the co-receptors that bring it over to the synapses, then you're going to have the actual transduction of the signal is going to be tamed, and that would allow you to have self-reactive cells that are not driving autoimmunity just on their own, right? Because I think that's a question that I was thinking to myself, that how do they keep these cells in check? And to which I would ask as a well, I guess you're already answering my question. These are all mostly CD8 alpha alpha, so you're not going to have regulatory T cells within this population, do you?
No. So we asked the same question. How can you have self-reactive T cells in the epithelium and not cause autoimmunity? And so what we did was we knocked out Tregs using the FOXP3 DTR mice. And we said, okay, well, what will happen? And you see that T cells get activated all over the body, but they don't get activated in the epithelium. So the epithelium is really not, the IEL and the epithelium are not under the control of Tregs. What instead we found was that the TCR signaling pathway is completely rewired in these autoreactive T cells. So they really can't signal very strongly. They have a lot less of one of the key adaptive proteins called LAT. They express a lot less of it, and they also express many other different changes, which makes them just really poor at signaling through their T cell receptor. So it looks like they are self-tolerizing. They have their own, directly in the thymus already. When they go through that really strong agonist selection in the thymus, they get rewired. They express CD8 alpha alpha. They express the chemokine receptors and the addressins that direct them directly to the gut. And the moment they leave the thymus, they already are no longer strongly self-reactive.
So do we know if they interact with Tregs then at all? Is there like some crosstalk? Is it a redundant function? Are these like before does evolutionary biology say where these suckers come from at all? Like, are they been around for a while and then Tregs came afterwards or something maybe?
That's a great question. I don't know whether we know the answer to that, honestly. What I can say is that, there's in a mouse model of CD4 T cell transfer colitis. So where you take rag knockout mice and you transfer CD4 T cells to rag knockout mice, they get strong colitis. If you transfer Tregs together with the naive CD4 T cells, they won't get colitis. If you take TCR alpha beta CD8 alpha alpha IEL and transfer them together with the CD4 T cells, they also won't get colitis. So it does suggest they are regulatory T cells in their own, by themselves. They don't need Tregs. They are regulatory T cells themselves.
That's intriguing. I like that. I mean, everybody's trying to take poor T-Rex's job. Everybody's doing all these things. Why cannot they just keep their own lane? Every cell is moonlighting as something else. There are, weird variants. T cells or lymphocytes in general are so extraordinarily flexible in their job description. It really amazes me to all these stories, especially IELs, but also ILCs and all the things they do, all the cytokines they respond to, all the how they migrate across the body, sending signals. It's just fascinating, and I'm really mind blown by all the diversity of T cell career.
I think we have to think about everything as a spectrum. We have to have these overlapping spectrums.
Yeah.
If everything just stayed in its lane, we wouldn't have the perfect protection, or pretty much perfect protection we have right now. As it is, things still go wrong. But I think there are a lot of fail safes in the organism level, right? So you have the ILCs, you have the Tregs, you have the IEL, but you have also the whole innate cells that then myeloid cells that then go and get the real proper adaptive immune system, which I don't think IEL are. They're not and then those T cells come in and they also are there to protect the gut. So it's many, many, many levels of protection.
All right. Well, I think our T cells are getting a little exhausted today. So before we end the chat, we always try to ask a non-science question about the scientist behind you. If you could have one superpower, what would it be and why?
So if I could, I would really like to understand how a cell thinks. I think that would be the coolest thing, because I come from a signaling background. I'm a biochemist. But when I come to immunology, I find T cells they have so many different inputs coming in, and they make so many different decisions. And I wish I could think like a cell. And that would be the perfect superpower, if I could understand how the cell thinks.
It would be overwhelming. Imagine. I mean, and of course, we are very keen, I think, in immunology. Maybe from your cell biology background, it's a bit less so. We're very keen on anthropomorphizing the cells and thinking, what is the T cell going for? What is thinking? What it's driving its program? And does it want to, leave the thymus and be special? But it is really a lot that goes on in that command center inside of T cells and immune cells in general, that it's really hard to study. That's what there's a whole
Exactly.
discipline, right?
If I was on a soapbox, it would be the one thing that I think immunologists need to do more of is signaling, to understand how all the signals are integrated inside the cell.
Yeah, indeed. Would you like to think like an immune cell, Jason?
No, I'm going to think like an enterocyte, but that's almost
Same thing.
Same thing. Stuff comes in, stuff comes out.
It's just hungrier.
Yes, like a macrophage.
But as I was telling you, we have now data that the IEL tell the enterocyte what nutrients to take in and when. So if the T cells, it looks like the T cells in this case have the upper hand still, because they seem to be telling the enterocytes based on the diet, based on what nutrients are coming in, what transporters they should upregulate. And I think that is so cool.
Wow, that's pretty cool. That's pretty interesting. See, in the end, T cells always running the show. Am I surprised? No, that makes complete sense. They are the smartest cells. If you want to think like a cell, it would be a T cell because they clearly know what they're doing. So on that note, end of story. We can finish our conversation right now because I think this is as good as it's going to get. But thank you. We had with us Dr. Mahima Swamy from the University of Dundee in Scotland, right? Yes. And talking to us about the fabulous world of the intestinal epithelium and the cells that live deep down in it. Thank you for joining the Immunology Podcast.
Thank you so much. It's great chatting with you all.
Thank you for being here.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at immunologypodcast.com to get show notes, including episode summaries and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast, via Blue Sky at immunologypodcast.com, or via email at info@immunologypodcast.com with feedback or to suggest a guest. See you next time.